Welcome back, friends. We've been talking about the immune system and immunology lectures in the series of videos. And in the last two videos, we talked about the cell-mediated immune system in the innate immunity portion. That is how once the pathogen infects us, the cells start protecting against that thing. So macrophages start engulfing that, interleukins are generated, inflammatory response is generated, and then the leukocyte rolling leukocyte is brought into the action, into the exact area of damage from the red blood cells and from the vessels. We saw that. Now in this video we will be talking about the complement system, which is another integral part of innate immunity, but in this case the complement system work in a humoral fashion. So this is not a cell mediated immunity, because till now we talked about, in last two videos we talked about the cell mediated immune response, but this is one very important kind of humoral mediated immune response, which is non-cell mediated, where we see the involvement of some protein factors in the whole process of the immune system and immunity generation. In the complement system means complement is nothing but protein molecules. These are protein molecules. Okay, there are different set of proteins that we know of, multiple protein molecules that is always present in our body, which is getting activated always in our body. Now at the time when I am giving this lecture, complement is active in my body. If there is any infection, it is fighting against that infection. So it's always on. The whole idea of complement is that it is always ready for the function. Okay. Now in the process of complement system is that idea of complement activation. Complement activation. Complement activation means when these complement proteins are getting activated, ultimately those proteins start creating pores in the bacterial body. Okay, so first they attach to the bacterial surface and they start creating pore. So as they create the pore, all those materials from the inside the bacteria will come out. It will do the osmotic imbalance, electrolyte imbalance, and as a result of that the cell ultimately die. That is the idea of complement activation in the first scale, killing a bacterial cell by creating pores known as membrane attack complex, okay, or MAC. So they form membrane attack complex and create pore into the target cell and kill it. Now before activation of this complement, there are several rounds of so many complicated interactions that must go on. It's an amplified process and kind of a cascade of reactions that should be continued to finally activate a complement. Once the complement is active, it can do the process of killing the cell, but before its activation we need so many different cascade of reactions. So there are three different types of complement activation mechanisms in our body. One is known as the classical process of complement activation, another one is known as alternative process of activation, and then the MBL pathway of activation. Now mainly we are talking about the first two major type of complement activation pathway, that is the classical pathway of complement activation and the alternative pathway of complement activation in this video. Now we'll just contrast between both the pathways. Well, ultimate goal is the same, production of membrane attack complex and then ultimately creating pore in the bacteria and killing it. But the approach that they provide to finally get that goal is different for both these things. I think we draw a straight line, yes. So let's start with the classical pathway here. So I told you there are cascade of reaction should be carried out, and the first thing about the complement is that for activating a complement you need the presence of antibody. An antibody should surround your target cell. For example, the target cell is bacteria, so antibody will be required which will cover up the target cell. Okay, it will cover up the target cell completely. So this is the idea. So once they cover up the target cell, which is known as the opsonization process. Now covering a cell with antibody means two things. One, is other cells which are phagocytic cells, for example macrophage, dendritic cell. They increase their appetite for all those cells which are covered with antibodies. So if a bacterial cell is covered with antibody, then those macrophage, dendritic cell will come and engulf that cell, engulf that antibody coated cell. That is known as opsonization. This is one of the important thing about the antibody coating. Second thing, if a target cell is antibody coated, complement can be fixed, complement can be activated. In that case, MAC will form, kill the cell. So ultimately, if you are coated with antibody, that means you will be gone. Either way, opsonization or with the help of this process of complement activation. Now if I look at here in the complement activation, what happens? Let's say this is the target bacteria and antibody is surrounding this bacteria, okay, and it is destined for the process of complement activation. So the first protein of the complement, because I told you that there are several proteins known as complement proteins. Examples of those proteins are written as C. C stands for complement. C1, C2, C3, C4, C5, C6, C7, C8, C9. So there are so many complement proteins, mostly up to the 10 that we see, that we'll be seeing here in this two examples, that are involved with so many proteins, and they involve. And all of those proteins have one thing in very common, that they have two subunits, subunit A, subunit B. And most of these proteins contains the B subunit is little bigger, A subunit is smaller. And the A subunit act as an anaphylatoxin, right? Act as a like helping in the anaphylactic reactions, okay? And subunit B, the large subunit, and that B unit is mostly attached to the cell of the target, okay? That is the idea. There is one exception, that is only for the complement protein 2, while the A subunit remains attached, B is released. But rest of them case, the other things there. So what happens in classical pathway? Target cell is coated with antibody. Then we have the C1. Now C1 carries three different structural units, Q, R and S, okay? C1Q, 1Q and 2R and 2S units. So this C1 is the very first and initiating complement protein which will be involved with the Fc region of the antibody. So C1 will bind there. Let's say this is the C1 with C1 QRS, 1 unit of the Q, 2 unit of R, 2 unit of S. So after the attachment of C1, they will recruit the second complement protein, that is C4, okay? So C1 recruits the C4 complement protein there. Remember, C1 is attached to the Fc portion of the antibody. Now C4 comes in, C4 in contact with the C1, C1 will break the C4 down into two parts, C4A and C4B, remember. Now C4B will be a part of the bacterial surface. C4B is a larger subunit, while C4A is a smaller subunit. C4A will be released, it will act as an anaphylatoxin, and C4B will be associated and attached to the surface of bacterial cell. Now second, this C4B will now break down C2. C2 will be broken down into again two parts, C2B which will be released and C2A which will be associated now and attached to C4B. Now they will recruit the third thing, this C4B and C2A together, C4B, C2A and C1 at the very beginning, which is the first to start with, but now we have C4B and C2A. Now the C2A and C4B together is acting as C3 convertase. So this C3 convertase is consisting of C4B, C2A. Now this C3 convertase can break and C3 down into two different fragments, right? Again it will break them down. So we have C4b, C2a as a C3 convertase. C3 comes along, C3a is released, C3b is now present. So now we have a complex of C4b, C2a, C3b. So see how it's going? Then this complex together, C4b, C2a and C3b, is known as C5 convertase. Now C5 is taken, again same thing, C5a will be released outside and C5b will be associated, C3b, C5b. So ultimately what complex we formed at the end? C4b, C2a, C3b and C5b. All the B components together, they are attached one after another in this surface of the bacterial cell. And while they do that, this is first step of reaction which is very important, where they form the C3 convertase, where once they are breaking down C3b, they start depositing lots of C3b on the surface of the target bacterial cell. This C3 convertase breaking down a lot of C3 and attaching C3b in different regions of the surface of the bacterial cell. Why they do that? The C3b, once surrounds a bacterial cell, can help in the process of opsonization. Remember I told you what is opsonization? Opsonization means once the cell is covered with either antibodies or the presence of C3b, it will recruit the process of phagocytosis. So phagocytosis will occur more frequently if it is opsonized. Okay, that is the idea. But ultimately, when this complex activates and produces this 4 complement system complex, this complex is now recruit some more complement proteins, that is complement protein 6, 7, 8. So they will attach with C6, C7 and C8. And ultimately, when they attach with all these complements, remember, so everything is attached to the surface of the membrane, all the complements together. Now finally, they will recruit the C9, and C9 will start creating a channel or pore in the membrane. Remember, up to this area, when they form C4b, C2a, C3b and C5b, they are not dangerous, but when they are associated with C5, C6, 7, 8 and 9, all of these last 4 complements along with this complex together will form membrane attack complex. Remember I told you, whatever type of complement system you are looking for, ultimately they are looking to form membrane attack complex, because if they form membrane attack complex, in that case it will punch the hole in the bacterial cell and the cell will die. Here in the classical pathway, first C3 convertase is activated. It will activate C5 convertase, which is this one, which will convert C5 into the more complex. Once this complex is associated with C6, 7, 8 and 9, morely 6, 7 and 8, they will form the membrane attack complex, and they will recruit C9, which will create the pore in the membrane through which all those electrolytes can pass by, osmolarity will be altered and the cell will ultimately lead to the death. That is the process of the killing of bacteria using classical mode of complement. You see the whole process of classical mode started with C1 recruiting C4, C4 again breaking down C2, complex is formed, C3 convertase using C3 breaking down, C5 convertase formed, break down C5, membrane attack complex formed, bring C9, create the pore, cell is dead. So this is the sequential events. While in the alternative pathway, if we talk about here, in the alternative pathway, they uses and rely more on C3b or C3 complement compared to the rest of them. The idea is this C3b is very important one, because if you C3 is a very important one, because if you break C3 down, you will form C3a acting as anaphylatoxin and C3b, which will be associated and helps in the process of opsonization. So both of them are important mediators and do some sort of signaling that can be visible from outside. Now this idea for C3 is that once C3b is formed, say the C3b is soluble because this is soluble complements. So soluble protein C3 broken down into C3b, it's still floating there. Now it will recruit one specific factor known as factor B. Factor B. Now C3b will be associated with factor B. For example, let's say this is the surface of a bacteria, and here we come C3b. C3b is now attached with this factor B. Okay, but this factor B and C3b, both of them are in inactivated form. Once this factor B is attached with another factor known as factor D, this is B. So once this factor D is interacting with factor B, it makes this C3b activated, and then this C3b is embedded into the membrane. It's then embedded into the membrane. Now what else they will do? They will now recruit more factor B, more C3. So now what they can do, they don't need any C3 convertase, that is C2b, C4b, C2a, they don't need any of that. They just recruit C3. This C3b will go, factor B will be bound to the C3, D will activate factor B, and C3 will be broken down to C3a and C3b, and the C3b portion will be attached to the first one. So as they initiate the process of embedding, they don't need any C3 convertase at all here, because in this case, to produce a C3 convertase, we need C4b, C2a as a complex. Here we don't need any other complements, only C3. We start with this process. At the very beginning point of this whole process, some of the C3 gets activated, gets destroyed and destabilized into C3a and B, and by that fashion, once they produce a C3b and embed it, then after that they will take just two factors, factor B and D, while C3 is interacting with factor B, D is activating the B, so breaking down C3, because B can break this protein down, help in the hydrolysis process. And remember, this whole process to occur, we need water in the first place for the hydrolysis reaction. Now every time C3 is there, B is going and attached to the C3, D is coming and activating B, which is breaking down C3 into C3a and C3b. C3a is released, C3b is attached to the membrane and embedded to the membrane. By that fashion, more and more C3 is brought, more and more C3 is broken down into C3a and B, more and more C3b is embedded to the membrane, and more C3a is released outside. So by that fashion, the more C3 will be there, the more C3b will be covering this whole bacteria. Now what is the idea? As more and more C3b is covering the bacteria, there could be two options and two fate for this bacteria. One is that proper opsonization, which will help them to be engulfed by the macrophages or the phagocytic cells. And second thing, this C3b can easily recruit other proteins like C5, because this C3b act as a C5 convertase. So they can convert this C5 into C5a and C5b, and it can go and revert back to the process and formation of membrane attack complex, and again killing the cell by forming membrane attack complex. So this is the idea of alternative. In the classical pathway, there is more complex, started with C1 recruiting C4, 2 to make C3 convertase, then recruiting some more to convert them to C5 convertase, and then do the rest of the job by creating membrane attack complex. While in alternative pathway, we just first need to produce a C3b that should be embedded in the membrane. To create the first C3b which is embedded to the membrane, it requires water for the process of hydrolysis of C3, and then they will bring this factor B and factor D. While D can activate factor B, and once factor B is active, it will break down any complete C3 into C3a and C3b. While C3a will be released, C3b will be embedded to the membrane. By that fashion, we can produce more and more C3b which are embedded to the membrane, and that circulating C3b in there can help in the opsonization. It will also help in the process of killing the cell with the formation of membrane attack complex, because any time the C3b can act as a C5 convertase and go back to the classical pathway. So that in a sense is the difference between the classical pathway of complement activation and the alternative pathway of complement activation. But remember, in either of this way, we see the cell is dead, because that is the idea, to kill the target bacterial cell, and that is how they achieve this task. But remember, for this process we need an antibody which is specific, which require little time to produce. But normally a specific, this is not required a very specific antibody there, because body knows what kind of bacteria usually infect us, and in those cases they produce all those immunoglobulin Gs and very common antibodies that are circulating our bloodstream. Those antibodies are covering the target cell, and we recruit rest of the complements which are also found in our plasma. It's not the part of the cellular process of the defense, present in the plasma, and recruiting them together we can kill the target bacteria either with classical pathway or with alternative pathway. So that in a sense is a complement system and how complement works. I hope this video helps you. If you like this video, please hit the like button, share this video with your friends, and subscribe this video to get more video lectures. Thank you.